Hello, everyone, and welcome to our module on acid excretion. There are two types of acids that the body produces every day via metabolism. The first type is called a volatile acid, and the second type is called a non-volatile acid. When we talk about volatile acids, we're talking about carbon dioxide. As you know, carbon dioxide is produced by metabolism, and it can combine with water to form carbonic acid and protons. And the way the body handles volatile acids like carbon dioxide is via the lungs, not the kidneys. Non-volatile acids, on the other hand, are not produced from carbon dioxide. They are derived from the metabolism of amino acids, fatty acids, and nucleic acids. So, for example, sulfuric acid is one of the non-volatile acids produced from metabolism, and sulfuric acid can potentially give off a proton, and this could significantly lower the pH of plasma. But this doesn't happen because we have bicarbonate in our plasma to act as a buffer. So, any sulfuric acid produced from metabolism will combine with bicarb to form sodium sulfate, carbon dioxide, and water. The carbon dioxide will be eliminated by the lungs, and the kidneys will eliminate the sodium sulfate. And so, because of the presence of bicarbonate in the plasma, acids are buffered and there's no change in pH. But note that this reaction on the screen consumes several molecules of bicarb. So, the bicarb has to be replenished by the kidneys, and that's the kidney's role in acid base balance. So now let's talk about how the kidneys regulate acid-base balance. The kidneys need to do two things in order to maintain bicarbonate as a buffer in the plasma. First of all, they need to reabsorb all filtered bicarbonate and also generate new bicarbonate, and then they also need to excrete H plus protons. We'll talk about both of these in the next few slides. So hopefully you're getting the picture now that the kidney's job is to regulate the bicarbonate concentration in the plasma so that that bicarbonate is available to act as a buffer for non-volatile acids. In order to maintain a normal bicarb level in the plasma, the first thing the kidneys want to do is reabsorb all the filtered bicarbonate. You don't want to let any bicarb go out in the urine. You want to recapture it all. And then once you've recaptured it all, the kidneys will worry about generating new bicarbonate to replace any that was lost in buffering acids. So most of the bicarb reabsorption in the kidneys occurs in the proximal tubule. The process involves secretion of protons in exchange for sodium ions. Those protons combine with bicarbonate, which is filtered by the glomerulus, and it is converted into carbonic acid. The enzyme carbonic anhydrase then converts carbonic acid into CO2 and water, which cross into the proximal tubular cells. Inside the cells, carbonic anhydrase converts the carbon dioxide and water back into bicarb and protons. The proton is then recycled back to the urine so it can be used to recapture more bicarbonate. The bicarbonate then goes out into the interstitium and blood in a pump with sodium attached. What this means is that this entire process has taken filtered bicarbonate and pulled it into the interstitium and blood so that the kidneys are reclaiming all of the filtered bicarbonate. And about 80% of the bicarb reabsorption occurs in the proximal tubule. Smaller amounts occur in the thick ascending limb, the distal tubule, and the collecting duct. And usually you don't excrete any bicarb, you recapture all of your filtered bicarb. Once all the filtered bicarbonate has been reabsorbed, the next job of the kidneys is to generate new bicarbonate to replace any that was used as a buffer for non-volatile acids. This process occurs in the collecting duct. In the intercalated cells of the collecting duct, carbon dioxide and water is combined via the enzyme carbonic anhydrase. This forms a molecule of bicarbonate and a proton. The bicarbonate is reabsorbed into the interstitium and blood, and thus the kidneys have generated a new molecule of bicarbonate. Note also that this process generates a proton. In other words, it generates acid. That proton must be excreted in the urine. If it were not excreted, it could recombine with the bicarbonate and negate this entire process. Because the generation of new bicarb creates protons, it leads to a high concentration of protons in the urine. This can lower the pH, and if the pH gets low enough, it can damage the nephron. For this reason, the urine needs buffers which can soak up the H plus and protect the nephron from low pH. In the plasma, as you know, we use bicarbonate as a buffer, but there's a problem. In the kidneys, all the bicarbonate has been reabsorbed by the time the urine reaches the collecting duct. Therefore, we need other buffers to serve in the role that bicarbonate plays as a buffer in the plasma. There are two important urinary buffers that are used to buffer the protons that are secreted from the process of generating new bicarb. The first one is called titratable acids, and the second one is ammonia. So titratable acids are urinary substances that absorb protons. They are acids and they are measured by a technique called titration. That's why they're called titratable. Most of the urinary titratable acids are phosphate, and phosphate can exist in multiple states. It can exist with one hydrogen attached, or it can exist in a dihydrogen form. The way phosphate functions as a urinary buffer is the monohydrogenated form is filtered by the glomerulus. It then forms the dihydrogenated form by addition of a proton, and that dihydrogenated form goes out in the urine, and when it does so, it is carrying a proton. In other words, it is carrying acid with it. What this means is there is increased excretion of the dihydrogenated phosphate form anytime the kidneys are excreting more acid. And here's a picture of how this whole process works just to make it very clear in your mind. When bicarb is generated by the kidneys, a proton is created as part of the process, as we saw previously. That proton is pumped out into the lumen of the nephron. There it will find phosphate molecules and combine with them, and the phosphate will carry the proton out in the urine. The second urinary buffer is ammonia, and the reason we need ammonia is because we have a limited supply of titratable acids. The supply of titratable acids varies with dietary intake, especially dietary intake of phosphate. So if the kidneys need to excrete large quantities of acid, titratable acids are not going to be sufficient to carry those protons out of the body. The supply of ammonia, on the other hand, is adaptable. The kidneys can generate more ammonia when they need to secrete more acid, and this makes ammonia a very important urinary buffer. Ammonia is synthesized from glutamine, an amino acid. Shown at the bottom of the screen is the structure of glutamine. It can be broken down to form two ammonia atoms. Now, just a little terminology here. Ammonia is NH3. Ammonium is NH4. It's an ammonia molecule that has picked up a proton. The process by which ammonia serves as a buffer is similar to that of titratable acids. Bicarbonate produced by the kidneys is pumped out into the interstitium in blood. The proton created by that process is pumped into the lumen of the nephron. There it finds ammonia molecules. It combines with them to form ammonium, and the ammonium shuttles the protons out of the body. What this means is that ammonia plays a role in acid excretion and bicarbonate generation. It also means that anytime there is increased ammonium excretion in the urine, the body is excreting more acid. So here's a summary slide of renal acid base handling. Non-volatile acids are produced by metabolism. They are buffered in the plasma by bicarbonate. This leads to a reduction in the amount of bicarbonate in the plasma. The kidneys want to reabsorb all the filtered bicarbonate. Most of this happens in the proximal tubule. A much smaller amount happens elsewhere in the nephron. In the collecting duct, protons are excreted and bicarb is generated to replace the bicarb that was used as a buffer in the plasma. As a result, titratable acids and ammonium go out in the urine. And a very high yield point to remember about this whole process is that anytime the kidneys are excreting more acid, they will excrete more titratable acids and ammonium. You're often asked about what the kidneys are doing in the setting of an acidosis, and you need to be aware that the kidneys are going to modify the amount of titratable acids and ammonium that's excreted in order to try and compensate for the acidosis. If you want to know how much sodium the kidneys are excreting, it's very straightforward to determine this. You do a 24-hour urine collection and you total up the amount of sodium in the urine. It's not so straightforward for acid because acid is excreted in multiple different forms. In order to determine how much acid the kidneys are excreting, you have to calculate something known as the net acid excretion. To calculate this number, you add up the urinary acid production over 24 hours and you subtract the urinary base excretion. A positive value indicates acid is being excreted. So to determine the net acid excretion from the body, you do a 24-hour urine collection, you total up the amount of titratable acids and ammonium, and you subtract the amount of bicarb. This gives you a number for the net acid excretion, and that tells you how the kidneys are responding over a 24-hour period in terms of acid-base balance. And the way this comes up clinically is that in a setting of an acidosis, you would expect increased net acid excretion by the kidneys. In the setting of an alkalosis, you would expect decreased net acid excretion by the kidneys. And that concludes our module on acid excretion.